Okay, thank you very much for inviting me to the conference. And before I start, I'd like to thank everybody who has bared me in mind to invite me to talk at this conference. In particular, Professor Luciano Silva. I thank him for getting in touch with me and inviting me to participate in his conference. So the work that we do in the biophysics group at University College London is to try and regenerate and reconstruct tissues for human implantation or transplantation. And what I'm trying to, and what I'm going to show is the work that is carried out in my lab for the last 20 years or nearing 20 years. And I'm also going to give you a bit of a brief history why we find ourselves with the problem that we have to try and reconstruct tissues and so on and so forth. So let me start my presentation and build the scientific case for it. So people are living longer, as you can see, and this is due to the advancements in modern medicine that have done some wonderful, had some wonderful advancements. These advancements have seen humans living longer. Although that being a good thing, the problem is that we are living longer, and therefore the body's tissues are deteriorating or becoming dysfunctional as we age, which is expected. And therefore this is causing a problem where there are much more older people in the system, if you like, than having younger people pay for the healthcare. And this is sort of the very, very brief socio-economic problem we see ourselves in today. So in this effort, obviously with tissues dysfunctioning, not only tissues but organs, there is a donor pool. The unfortunate thing about the donor pool is that the demand is always outweighing or overweighing the supply, and this is causing a lot of problems. For example, some of the tissues that or organs that we get are not fully functional, and they cause problems for the patient that it goes into. As you can see from my presentation, you can see that some patients unfortunately pass away because of the wait, for longer waiting times. But that being said, this is the first gentleman who has created, contributed to this. This is Alexis Carrel, the gentleman you see on the left of your screen. Now he's the person with Charles Lindbergh, as you can see on the front cover of the Time magazine, who funded his work and who supported his work. And what he did was, Alexis Carrel showed that you are able to actually culture cells outside the human body. I mean, there are a bit of controversies. You'll read about that, how he ran his lab and all that sort of thing. But regardless of that, Alexis Carrel was the first to actually try to develop or grow human cells outside the human body. In fact, that was his book that he published, The Culture of Organs, and with together with Charles Lindbergh, who you might have heard of. He's an aviator who crossed the did some very notable flying exercises, and as I said, he was a support towards Alexis Carrel. Now, along the path, the road to tissue engineering or regenerative medicine, these two gentlemen, Joseph Murray and Donald Thomas, actually contributed a great deal by doing the first renal transplant. And I should also mention and make note that everyone here at the presentation also are aware that both Alexis Carrel, Joseph Murray, and Donald Thomas all have been awarded their Nobel Prizes, obviously not at the same year, obviously, but at different years. And the reason why the two gentlemen that I just showed you were awarded the Nobel Prize was because they were able to do the first renal transplant. Since the transplants were done, as I mentioned, that transplants transplants can be done and can be very successful. That being said, as I said, the demand is outweighing the supply. Regardless of that, bearing that in mind, Weinberg and Bell was the first to show that you can actually engineer an actual vessel. And the article that you can see published in 1986 in Science, this showed that you are able to actually try and build an actual a tissue or a structure outside the human body which could be possibly used for human transplantation. Subsequently, in 1990, this scientist showed that by taking given cell types, putting them together, you can actually create sort of the mammary glands or mammary tissue that were shown to actually provide or create milk. So these two papers are quite very significant, because one shows that you can actually build a structure with collagen or various other biocompatible materials. And this scientist here showed that you are able to take the cells and actually build a culture by mixing the various sorts of cells found in a mammary gland or a mammary tissue, and then you can create for them to actually build an actual tissue that is that mimics the actual native tissue found in the organism. Subsequently, there have been, this is very recent, subsequently, and I'm sure you have heard in the news that a whole community of scientists are trying to knock off certain genes of animals, in particular pigs, and try and build human organs or human compatible organs in pigs, which could be used for transplantation. Unfortunately, that although the concept and the idea is very young, and there's a lot more advances that will come along, the recent transplant of a heart to a human, I gather did not, the transplant was successful, but the patient didn't live long, and in fact after a couple of months, I guess, or I think, the patient passed away. The other approach people have been taking is to actually take a donor tissue and tease away all the cells, put the cells of the patients into the tissue, and then transplant it. And the reason they do that, or the thought behind that, the school of thought behind that, is that in the case of donor tissue or donor organs being put into another human, the tissue actually is rejected, and therefore the patient requires lifelong immunosuppressants. Now, in this case, where you can actually take a heart, tease away all the patient's the cells of the donor out of the tissue, where you're only left with, as you can see, the third image of the heart, which is completely stripped of all the cells. Then you start giving it all the adult cells from the patient that it is going to go into, and repopulate it, and it'll start to beat as expected. That being said, this once again is much like the donor concept. It is taking one step forward, in my opinion, and then taking 2 steps or 3 steps back, because you're once again relying on the donor tissue. And I gather that this approach has a lot of controversies with regards to the chemicals that are used to tease away the cells are compromising the integrity of the collagen scaffold that's fully cleared. And even if it is fully cleared, there may be DNA and RNA residues that could cause issues or sort of immune reactions. There have been other approaches where the, sort of structures have been created outside the body and then injected with various cell types, and then surgery is being carried out, and replacements of these tissues or reconstructs generated outside the human body are actually transplanted. Unfortunately, that has been very unsuccessful. And this article now has been retracted, and there's a whole bunch of controversy about that as well. So you can actually see what the state of the art, what is happening is that people are trying to actually use tissues from animals, from donors and various sources, but the unfortunate hurdle that they can't seem to overcome is the immune rejection and the actual ability to have a fully functional three-dimensional tissue that mimics or is indistinguishable with native tissues. Now, this particular group has taken the idea of actually building a scaffold and seeding cells onto it, much like not taking an actual human tissue and decellularizing it, but creating a scaffold and then repopulating it with the cells. Although this, as you can see from the image here, what you're doing is you're taking the actual scaffold, repopulating it with the cells, keeping it in a bioreactor for many, many months or a year, and then reimplanting it. The unfortunate thing is there are clinical studies being carried out on this approach. The unfortunate thing is the waiting times are very, very long. And therefore, the concept of on-demand tissues really is not on demand, if you like, or in my opinion anyway. Now, the problem that we see, as I mentioned to you, is that although there have been a lot of advances since the time of the early contributors, such as those I mentioned, Alexis Carrel and the others, we still grapple with the ability to grow cells in large quantities, to have advanced vascularization. For example, you can build the best tissue in the world, but if you can't vascularize it, or if it doesn't have vascularization, the tissue is going to die because there are no nutrients supplied to it. Biomaterials play a critical role, and we are always having biomaterials that are developing as we go along, and there are new biomaterials coming along. Unfortunately, this, much like many other the process that we have to follow, which is there for good reason, takes 10 to 15 years for it to actually show up for clinical utility. And there are various other little issues that moving from in vitro to in vivo, and then large scale going to human scale. And there are several issues like that as you move on. Now, in my presentation, I'm going to identify two different ways of building a tissue, and they are namely direct and indirect methods. The direct methods is where you take all the constituents found in a tissue, for example, which is 3 to 4 types of cells, add it with various cytokines, proteins, collagen, and all the constituents found in tissue, as you would find if you dissociate it, you bring it all together and then you deposit it, for example, using a printing technology or some form of molding technology or whatnot. The indirect method is where you would actually imprint sort of proteins and then you call cells to the site. Unfortunately, the former is better than the latter because the number of steps taken to achieve the development of a tissue is much less time. And there are various other issues. I will go through them as I go through this presentation. So there are all these approaches. These are some of the approaches I chose to highlight. There are some retrofitted technologies that you'll be, I'm sure all of you would have read or come across, but unfortunately, those either take many, many steps in getting towards the end goal, which is to build an actual three-dimensional functional tissue, as opposed to going through the process and having issues such as sterility issues and various other issues of scalability and the ability for cells to be processed without killing them and so on and so forth. So in the direct tissue engineering or in the direct approaches, the main approach that actually started this technology, or started this concept, is 3D printing that I'm sure you have heard of. 3D printing, which is that there are several subsets, for example, solenoid-based, piezoelectric, bubble jet, and various other approaches. And I will show you the shortcomings of those approaches and show you other technologies that have overcome them. The four technologies that I've highlighted in red are the technologies that we have in my lab pioneered, and I will be very, very critical of the technologies that I highlight in red, which are the technologies, as I said, we have pioneered, but also to compare it and argue it out, so that you, the audience, will able to decipher what is good and what is bad. So, as I said, in the direct cell jet-based engineering protocols, there is inkjet printing, which I said breaks down into, you go to the left side of your screen, you can see thermal, solenoid or valve-based, and piezoelectric. And then there's aerosol delivery deposition and lab on a chip. The unfortunate thing about inkjet printing or 3D printing that you read about in the popular press is that cells are actually, the processable amount of cells are limited, and they are damaged during the printing process. Aerosol delivery is very similar. The process is very harmful and very aggressive on the cells, and therefore it has effect. Lab on a chip, although is able to create wonderfully controlled cellularized structures, the yield is very, very small, meaning that you can do wonderful things to maybe to do experiments on a mouse, but if you start to do experiments on, say, a large animal and moving towards human, for example, primates and sheep and so on, the yield of the structures that you can create using lab on a chip fall short. Once again, I mentioned the technologies in red. I will talk to you as we move on, are the ones that were generated in my lab. Now, the problem with 3D printing, why it cannot be used for tissue engineering or processing cells, living biological cells, is that the number of the needle basically used in 3D printing technologies is very small, is about 30 to maybe 60 micrometers. Now, if you look at human cells, red blood cells are about 5 to 10 micrometers. So if you think about it, within the needle, if there's a parabolic flow pattern, and if cells are, if there are more than 6 or 7 cells across, as they pass through the needle, there will be shearing on the edges of the needle due to the parabolic flow rate, due to shearing. So this is the problem that creates cell death and even cell augmentation, which is not a very good thing. If you think about it now, the example I gave you is a single cell type, but if you put multiple cell types, because in a tissue you don't get a single cell type, you will have cell types of wide ranges. Say, for example, cardiac cells are about 100, 150 micrometers. So how can you put a 150 micrometer cell through a 60 micrometer nozzle? Okay. And if you increase generally in 3D printing, it's a general practical aspect that anyone who works with 3D printing will understand, that if the needle diameter is 30 micrometers, the droplets that they generate are about 60 micrometers. So the residue, once it's deposited, is about 100 micrometers. So the problem is that it can't process large volumes of cells, but if you increase the diameter in the nozzle, you're increasing the size of the residue. So what if you wanted to print something like a vessel in your eye? Okay, so bear that in mind. A lot of studies, since we've published our work, we've been very critical not only of our work, but of work of others, other technologies, and they've done FACS, for example, and very recently, and they've shown that 60 to 70% of cells are dead after the process, which is not a very good thing. So 3D printing, as you know, can build arches and various structures, but the problem is As you can see in the middle image, the blue part, you need a structure to hold it. So if you're printing biological structures, these cause increasing problems in sterility. As you can see here, you can see a bone structure is being printed, but the overhang, to support it, you've got to print this support. Also, what is misleading, for example, is that a lot of the work that has been in the popular press show that if you wanted a heart, you can easily print a heart, liver, or kidney. This is not true by any stretch of imagination, and this is at best misleading. You can see these are papers published by other international groups. You can see by 3D printing, you can damage proteins. You can see by 3D printing, you can also damage cells. Note in particular, the cells are damaged due to shearing, as I mentioned before, and these are when you process single cell types. Just imagine if you were going to print a tissue that has 2 or 3 or 4 types of cells in it, which generally is the case. And even if you were to put single cells in, what about the polymer that you mix the cells into? The viscosity of that polymer will also have effect on the cell death. So the ability to process large volumes of cells requires large bore needles, inner bore needles. But if you can find a process that you can use large bore needles, but also process and generate very fine droplets, that is the key to a successful process. And the ability to not only handle cells, but also to handle various materials, and also to be able to not only generate droplets, but also to switch to generating threads. The only process that is able to do that, and added to that, when you process cells, you must be able to use all these characterizing processes. These are just some characterization approaches, but you can, for example, RNA sequencing, whole genome sequencing, and whole transcriptome analysis are also essential, because we must, the processed cells must be completely indistinguishable with control cells. Otherwise, you'll be putting cells into the body that are genetically altered, and that's not a very good thing. So the only process there are 2 processes that can do this, which is to use large bore needles, but form very, very fine droplets or threads, and to be able to switch between the 2, and that is namely electrosprays and electrospinning. These technologies are not new. They've been around for about 100 years. And you can see that electrosprays work on the principle that the nozzle that it comes out of is charged, and there's a ground electrode placed somewhere below it. And the difference in charge from a charged needle to the grounded electrode generates the charged liquid that's flowing in the charged needle to accelerate in the form of the formation of a cone, from which the apex, a jet emanates and breaks into droplets. Now the droplets break up due to surface tension effects, and if you change the viscosity of it, you get threads, and that is where you get the switch from electrosprays to electrospinning. And you can see these images show that you can use a very large bore needle, inner bore needle, but the generated jets and the droplets are very, very fine. In fact, both these technologies are able to use inner bore diameters of about 1,000 to even 2,000 micrometers, yet generate droplets or residues in the few nanometers. And when I say few, in the sort of 5 to 10 nanometers. These are some of the images. You can see that they have processed and generated. You can see the sort of structural scale, and you can see the sort of structures that you can build and switch from droplets to fibers and scaffolds. There are various ways of fine-tuning these, and you can have not one needle, but 2 needles, coaxial needles, triaxial needles, and form structures like onion structures that have different compartments. So the versatility of this technology is phenomenal, and the production rates are very, very high. These just to show you that electrosprays and electrospinning are not new technologies. You can see that it goes into many, at least a century and beyond, as you can see. Now, in the form of, in tissue engineering, a lot of people, what they've done is formed scaffolds. As I said, then they place cells on top of that, and then they implant it. But unfortunately, that technology, you can see the cells only over the period of 7 days have only infiltrated, if at all, 100 micrometers in thickness. This is not very good, unfortunately, because of the simple fact that if cells want to go into the tissue, whether it is from the host or from the structure to the host, you must be able to have tissues that cells within the tissue that actually moves in and out of the host or from the reconstruct to the host as well. So these are the limitations that currently exist. And as I said, because these scaffolds are also very tightly knitted, they actually form, they actually hinder cell infiltration, and that sort of causes foreign body rejections. So I think, I mean, I'm not going to read off the screen, but I think if this is recorded, you can go through these and come back to me if you have any questions. By the way, if you do have any questions, please don't hesitate to get in touch with me. My email and my contact details are on the web. Now, electrospinning, I said, is able to form scaffolds and form very, very fine fibers. The electrospinning community has been generating scaffolds and saying that you can build a 3-dimensional fully functional tissue. We didn't agree with this, and we investigated this, and this was not true. Subsequently, a colleague of mine, Kim Woodrow, about 5 years after what we did with electrospinning, came to us and said that, have you seen what's this, the use of these scaffolds as materials for stopping sperm from going through. Now, sperm, if you can understand, is the smallest self-propelling cell. Now, if the scaffold using electrospinning can stop the smallest human cell that is self-propelling, then how can an adherent cell go through? So her work was published, as you can see here, and she has also got funding subsequently through a patent and various other wonderful wonderful funding agencies to form novel contraception-based materials. The work that we did, we actually showed that when you, after you make, we made 4 punch holes on the back of a mouse, and we inserted these collagen scaffolds, and we found that the actual cells from the host, from the injured host tissue, cannot infiltrate the scaffold because the actual scaffold that electrospinning generates is so fine, and the open cells in them are very, very small, so much so that even cells can't creep through it. And you can see here, and I'll show you the paper after that, and you can see that the top layer of the skin, although it forms, if you look at panel A here, you can see that the actual top layer of the skin just goes, or sort of over rolls it, so it doesn't go through it. And these are signs that there is an incompatibility issue. And we have also seen in our studies, subsequent studies, that foreign body rejections were taking form. This is the article that we published, and it was with my colleague David Becker, who's a well-known wound healing scientist who has done some wonderful work with wound healing. And we show that these scaffolds, electrospun scaffolds, are ideal for using it as contraception materials, but not as tissue engineering scaffolds. So the whole point of tissue engineering as we see it today is that we take the workflow, if you like, is that, this is very simplified, mind you, is that is to take the patient, a biopsy from the patient, grow the cells outside the patient's body, expand the cells to the quantities that you require, hopefully, and whatever cell type that doesn't expand in the quantities that you want, you bring in induced pluripotent stem cells or various other stem cells, and you start changing their unspecialized nature to those specialized cell types that require quantity in the tissue that you want. Now, what we have done is we were hoping to get in where I have indicated here, advanced cell engineering protocols, where we take all these cells, mix it with the polymer and whatever other constituents like proteins and so on, and then build a tissue and hopefully transplant it into the patient. So the processes have been where we take different cell types, multiple cell types, not only single cell types, but multiple cell types in large concentrations, and form a cell suspension and either bioelectrospray, cell electrospin it, or aerodynamically assisted biojet or aerodynamically assisted biothread them. Now, I told you that electrosprays and electrospinning have been, they're not new, they've been used for over 100 years, but we were the first to show that you can actually electrospray cells and electrospin cells and also aerodynamically assisted biojet and thread cells without killing them from a molecular level upwards. So these were our first studies that we electrosprayed the cells. You can see that although the jet has formed a nice cone, the actual jet is rather oblong in nature rather than being uniform, and the droplets, as you can see, the high-speed photograph there shows sort of a distribution. Laser spectroscopy showed us that there is a trimodal distribution, as you can see from the plot here, and that is because, excuse me, as you can see, the large two peaks on the right of the screen are the ones that contain cells. Every other cell type, every other distribution on the left side of that graph is a blank. Regardless of that, after we sprayed that, we published this paper where we repeated many times over, because we found that the cells don't die. And we were a bit perplexed as to why the cells won't die, because we were spraying at about, in the thousands of kilovolts, in the thousands of volts, and we were sort of perplexed as to why the cells don't die. And you can see here the sort of cells that we actually bioelectrosprayed initially were sort of Jurkat cells, and we also did a lot of primary human cells, and you can see here the cells are undergoing mitosis post-processing. We also further elevated the applied voltage between the needle and the ground electrode, and you can see sparks started flying, but yet the actual spray did not, the cells were not found to be dying. So we started looking into this, and unfortunately, Professor Berg, who I was in contact with, is no longer with us, but I reached out to him and I was talking to him, and he introduced the process well used by geneticists, which is electroporation, where you use a current of 55 milliamperes with a voltage of 230 volts, which is the opposite of what we use. We use voltages in the thousands of volts, but use the current of about 5 to 10, maybe 20 nanoamperes. And that is what saves the cell, as opposed to electroporation that kills the cell. It's the current that kills it. Because in electroporation, the current makes the cell's membrane permeable, where you get your genetic construct into the cell. Once that process has been done, some cells do repair themselves, some cells don't. And geneticists have shown, using electroporation, you can lose about 60 to 70% of cells, depending on cell type. And that is something that geneticists have learned to live with. And this is where we found that in electrospraying and electrospinning of cells, bioelectrospraying and cell electrospinning, although the voltage is very high, in the thousands of volts, the current is in the nanoamperes, and this is why cells don't die. That goes back to the point of using a taser gun, which is the taser operates at about 10,000 volts, but at a very low current. And that's why it doesn't kill its victims. You can see that initially we started electrospraying a wide range of cells. I should also mention that till today, We have sprayed over 600 to 700 different cell types, ranging from primary human to immortalized to primary and immortalized animal and various other cell types. You can see the sort of structures that we've been able to build, and you can see that we build cells inside capsules, polymer capsules, and you will see that this not only has the ability to build tissues, but you can build organoids and so on and so forth, because of the simple fact that unlike normal organoid generation approaches, the cell encapsulations here have spaced out the cells. Therefore, you can actually put in various other types of cells like vascularization and so on for them to take form. So electrics in the surgery, in the room, in surgery is not a new thing. And this is very important if you are taking this technology into the clinic, because if there is some prior art, it actually helps the process move quicker into the clinic. One technology that is already uses electric fields well in the clinic is electrosurgery, and this is used for cutting lesions, as you can see. So the questions what we want to address is that, can we get a near monodispersed distribution of drops containing the number of cells inside them? And after a few months, when we looked in the literature of electrosprays, we found that you can use coaxial needles, one needle within another. So the cell suspension has the properties of a highly conducting because of the requirement of high concentrations of ions to maintain the cell's metabolism, while also the viscosity is low. Two characteristics that generate instability in the technology because it's an electric field driven approach. So therefore, what we did was we had the cell suspension flowing in the inner needle, while the outer needle held the flow of a high viscosity, low conducting silicone oil, bio oil. And you can see here that we were able to get a near mono distribution, as you can see from the plot. And you can see, although there is a variation in size, as you can see, but the variation is not significant. This is the article that we published showing that you can get stability in the jetting process. You can see that we have looked at various ways of looking at proteins and viability using flow cytometry. And the reason for using flow cytometry and basically clinical readouts is that assay-based evidence is ideal for lab-based to convince colleagues who do a lot of biomedical lab-based work. But if you want to take something to the clinic, you have to show clinically relevant readouts like facts, sequencing, and so on and so forth, karyotypic studies, and so on. And these are some of the papers that we have published. I'm not going to talk all about it, but if you have any questions, please, if you read any of these articles and you have any questions, please contact me, and I will be more than happy to go through them with you. So we've processed cardiac cells, various cell types in not only single suspension, but in multiple cell types. You can see here we published where we can electrospin cells, and this is pretty neat because you can actually form a fiber that contains cells inside them. And I'll talk to you more about that as we go on. So we've not only processed single and multiple cell types, but we've also processed embryonic stem cells and whole organisms. For example, fertilized whole organisms. And this goes to show, you can see here that we've processed zebrafish and we have compared it with controls. And here we can see that the brain fold on the left of your screen, on the far left is the control, and just next to it is the bioelectrospray, and the cell electrospinning is very similar. But you can see that the brain fold and the brain development is not perturbed. This is very important. Additionally, being in the United Kingdom, we were not allowed to go into the development of the zebrafish longer than a certain developmental stage. Therefore, what we did was we electrosprayed and electrospun all these organisms, and I'll point you towards the paper. But as I said, living in the UK Home Office does not allow us to take, say, for example, the frog, Xenopus tropicalis, the zebrafish, as you can see on the top, and Drosophila through to a certain stage. Or I think it was only zebrafish and Xenopus tropicalis, the frog, but the fruit fly, the C. elegans, the worm, and Dictyostelium, the home office doesn't have a restriction, and we took them through the entire life cycle. And we were able to show that the cells or the whole organisms are not perturbed in any way from a molecular level upwards. These are the articles we have published. We have also published the handling of various other organisms, as I mentioned before. And the important thing is one of my heroes, if you like, in science, John Fenn, who got the Nobel Prize for electrospraying proteins into a mass spec for inventing what is well known now as electrospray ionization mass spectrometry, was where they electrosprayed proteins. Now, because we can electrospray cells, you will be able to electrospray cells into mass spectrometry and identify various alterations and various other characteristics. And there are groups now doing that. So how do we switch from when using a coaxial needle from electrosprays from droplets to fibers? That's pretty easy, because the outer needle, we just change the viscosity. And once you change the viscosity, you start getting fibers. And because the needles, the bore diameters are so large, and you can have coaxial and triaxial needles, you can start to put various constituents, tissue constituents, and various other additional enhancing features into these tissues. These were the first studies that were published in 2005, 2004, 2005 sort of time. And you can see that we electrosprayed living cells, electrospun or cell electrospun living cells, and you can see the fibers have sort of protrusions containing the cells. This was my first time I actually did fluorescent microscopy. So I'm a material science scientist by trade, so I was not very good at fluorescent imaging, but I assure you that I've got better. This was the first paper that we published on that, and we showed that you can electrospin, and we coined it as being called cell electrospinning. And since then, there have excuse me, there have been many groups that have used this technology for a wide range of applications. In my quest for getting better in fluorescent imaging, you can see that we have GFP'd the cells, and we have made scaffolds, three-dimensional scaffolds. And the beauty of this is that the cells are right the way through the entire thickness of the scaffolds. And these are hydrogels, so the cells can either come out or cells from the host can go in. So it is forming a complete three-dimensional scaffold. So you can undersee the applications, not only tissue reconstruction and repair or rejuvenation, but you can, if you like, freeze cells in 3 dimensions like this, or even culture cells in 3 dimensions. So you can see the applications are getting more and more. We were able to also submerge the needles and spray cells inside, and you can see using coaxial needles, you can have 2 cell types, 3 cell types, and actually create a wide range of structures for this. And this was a piece of work that we are doing with a colleague of ours at UCL with regards to spina bifida. And the reason for doing that is that we'll have to spray in the amniotic fluid. And that's why we developed this approach. We also did some other work with, you can see with human sperm and so on and so forth. We can actually spray human sperm and not damage them. And this is wonderful because we can actually not only better the way of storage, so the recovery is better, but also to actually sort of culture them and keep them in the environment that they are better used to is very important. These are the sort of structures, these are beads that contain the sperm cells, as you can see, and we can freeze them, we can make them have various materials that during the freezing and the thawing process, the cells are not damaged in any way. You can see here a video of a vessel that we created in sort of a Y junction, one having three different sizes with 3 different cell types. You can see red, yellow, and green. So you can build quite, advanced structures or complex structures using this approach. We can also bring in genetics into it. We work with genetics, our colleagues at UCL, genetic colleagues, colleagues who are geneticists, and we incorporated sort of experimental cells and our genes or medical experimental medical cells or genes into these encapsulations as beads or fibers, as you can see. And therefore, you can build a wide range of structures. Now, one of the applications that we have worked on very well is that we want to build three-dimensional tissues, models. And you can see here that we built these three-dimensional tissues as beads. And we went on to build, we modeled human tuberculosis. We were the first to do this with our colleagues who are TB experts. Professor Elkington, so one of our biggest collaborators, a wonderful guy who knows, who's experienced who has a vast experience in respiratory medicine, and he works on TB. So you can see that we were able to model TB. These are some of the papers that we published with him. And so you can use these structures with experimental cells or genes for, you can use your patient's own cells that are genetically altered. Therefore, the genetic payload is the patient's own cells. So, and you can see here that we have created organoids with vascularization taking form. And we have no problems in the problems that have been faced by hanging drop technologies and various other lab chip technologies. We don't face any of those because the production rates are high. And as I said, the cells are widely spaced rather than being clustered and causing hypoxia and central necrosis, etc. These another piece of work that we did with our colleagues in King's College London with Professor Modare and other colleagues, and we were actually able to show that we can trigger vascularization in the hind legs of a mouse. And this is very important because when you, I believe that when we get older, that we lose vascularization in our legs, and therefore this is one possible way that we might be able to combat as we take this through the process of development. We've also looked at various, I was also talking about assessing these cells, whether they are damaged or not, and we've done a whole slew of studies using well-established clinical readouts and assay-based readouts that are well established in both the biomedical lab and the clinic. And one such approach is where we actually took the spleen cells of a mouse, we electrosprayed them, and we injected it into the tail vein, and we found that the cell's ability to home and its specificity were not altered at all. We subsequently built tissues. As you can see here, we have built tissues such as implants in skin. We've seen the cells as a function of time, build 3D networks, and we have implanted them in mice. We've done various studies, as you can see, FACS, Brodo, and various other well-established assay-based studies and clinically relevant approaches to assessing the cell's well-being. These are some other studies that I'm just going to point you through to show you that we've actually moved from in vitro to in vivo. And then these are the mouse studies that we've done and shown that we are able to electrospray or bioelectrospray or cell electrospin and transplant without having any negative effects. As you can see, we've formed these platform technologies now, and we can actually build tissues not only for building an actual three-dimensional tissue, but it has very applications from human healthcare to, if you look at what is happening in areas such as advanced in our food industry, you can reconstruct in vitro meats using cultured cells. And you can see these are the tissues that we have actually sort of implanted and taken sections of and analyzed the actual cells and the tissues. We are also able to build vessels. As you can see here, these are vessels that we built. This vessel was using cell electrospinning, and we were able to build, currently we are able to build with the equipment that we have, are able to build a tissue of about 7 to about 9 inches in length. The hope is that we have submitted an application for funding to actually build a vessel that is about 7 feet in length, obviously, to mimic a vessel that goes from your brain right the way down to your feet. So we will be able to build that and keep it so that when you require it on demand, you'll be able to cut whatever lengths and sizes and use it for implantation. So you can see the classical school of thought of actually making a scaffold and then seeding it with cells, then going into the bioreactor, and then you will have to switch between manually seeding and going to the bioreactor off and on quite a few times, because the cells will need to infiltrate. And once it's ready, which will have to go through that process many times and then go into the human, we stop the number of stages by actually incorporating the cells into the fibers and building the vessels or whatever structure, putting it into the bioreactor for it to actually start creating its own extracellular matrix and so on, and go into the human. So the number of stages and the reduction in the form of sterile tissues is much reduced in the approaches of using bioelectrosprays and cell electrospinning. And as I said, you can use these technologies for building, not only preserving cells in 3 dimensions, but also culturing them in 3 dimensions. And at present, there is no approach for culturing cells in 3D, although various advanced cell culture flasks have been made, but they're really just 2D and not 3D. And you can see, if we are using the concept of decellularization, you can make your structure with your cells and then put it into a bioreactor and have them growing in a if you like, a cell bank, which is plausible because we currently have blood, if you like, in cell banks. And then when required, you can decellularize your own cells, if you like, out of them, and then recellularize it with your cells and then put it back into your body. So this is one approach of doing it, while for having it on demand, as opposed to taking a longer time and creating cells. So this is when a patient, for example, if a patient needs tissues right now, this approach could have your tissues in a bank. But if you were on the battlefield or something like that, you could electrospin cells straight onto the wound area. So there are you can see the flexibility of these technologies are sort of incomparable with any other approach. Various other technologies, we've applied these to various other technologies where we encapsulate these cells as we go through a growth factor. And then, for example, we do this with stem cells. And what we do is we create these beads with cells, and we tag them, then mix them, send them through a different growth factor, and then keep doing that, mixing and pooling. And then you can, every time it goes through a different tag, is tagged onto it, because you can, like I said, you can have like an onion structure, so that once it has about, I don't know, goes through about 5 or 10 growth factors. If you look at the structure, because it's completely see-through, so you can see the cells inside, and you can identify what cell types they are, if they are moving from unspecialized to specialized, and the tag colors will show you what growth factors it has been exposed to. So it's a combinatorial stem cell technology. This has been patented and is now in use in the industry. So the applications, you can see, are wide-ranging. It's medical diagnostics, cell therapies and tissue engineering approaches, to building organoids, to a wide range of you can build tissues and then screen drugs and vaccines and develop various new drugs and therapies and moving towards personalized medicine. These are some of the presentation slides. These are some of the special issues we've done. There are many more out there, and I urge you to, if you have any interest, to read them, and if you have any questions, please come back to me. That ends my presentation, and I'd like to once again thank Professor Silva and the entire organization committee for inviting me and bearing my name to come give a talk. Thank you very much.